I'm Cate Speake. I'm a PhD scientist in BRI's Center for Interventional Immunology. And my role is to work with people with and without immune diseases to make sure that all of BRI's important scientific findings get translated to the clinic, and that our scientists are able to study the kinds of samples that they need. Our team does a little bit of everything as long as it involves people, from enrolling participants in really big phase 3 clinical trials like the Pfizer COVID vaccine study that enrolled here at BRI, or asking people with rheumatoid arthritis to give a single blood sample to help us understand autoimmune disease. At the start of the pandemic, BRI was able to really quickly pivot to studying the virus. At first, we were able to generate a whole lot of data about the immune response to the virus itself, and more recently, we've been thinking about how the body responds to COVID vaccines. And in particular, we've been thinking about how people with autoimmune disease may respond to COVID vaccination, because we're in a really special position to have a community of people with these diseases who are ready to step up and help us. So the question that we had was this: people with many autoimmune diseases take therapies that modify the immune system, and this is really important in order to prevent the symptoms and prevent disease progression in those settings. But what we and a lot of our study participants also wanted to know was, do those therapies get in the way of making a full response to COVID vaccination? And what do I mean by a full response anyway? Well, in general, we want to see both a T-cell response and a B-cell response to our vaccinations. The B-cells are important because they make antibodies. Antibodies are able to prevent the virus from entering the cells that line the lung. T-cells are important because if any of that virus is able to infect the lung lining, the T-cell can destroy the infected lung cell to keep that virus from replicating. We've been really focused on trying to understand the B-cell response first, so asking, what does the antibody response look like in our population? Our team enrolled over 200 people in a study to try and get at this question, and to look at it in many of the populations that we study. This included people with rheumatoid arthritis and multiple sclerosis, who were each on one of many different therapies of interest. It included people with diabetes or Down syndrome, who have diseases that impact the immune system but typically aren't on specific immune therapies. And it included people with no immune disease to help us understand the variability in the general population. And what we found was pretty surprising and in general, very good news. We saw that many of the therapies that our participants took for their immune diseases didn't have much impact on their ability to make an antibody response. Their responses were generally in the same range as the controls, which was really good news. This was also true for our people with type 1 diabetes. We didn't see any difficulty in responding to vaccine in this population. We shared samples with collaborators at Yale for this study, and they showed that people with autoimmune disease didn't have an overactive response to the vaccine. So they didn't generate new autoreactive responses against their own tissues. They just generated a response to COVID, which is exactly what you'd want. The exceptions in our study were people who were taking therapies that directly impact the level of B cells. These are the cells that make antibodies, and so therapies that reduce B cells tended to reduce the ability of people to respond to the COVID vaccine. The big message, though, is that most people, including people on most immune therapies, were responding pretty well to their vaccine in terms of that antibody response. We're continuing to work hard on this study, and that includes looking at the T-cell arm with a special focus on people with rheumatoid arthritis and multiple sclerosis. It's really been a building-wide effort from the beginning of the pandemic. And that parallels largely, how it's always been here. We're all working together to address the causes and the consequences of immune diseases that impact our community. I think 2 years ago, none of us really knew that this is where we would be today. But I think it's really been amazing to be able to be part of the work that has helped us understand COVID and will hopefully help us get past it.